Have you wondered how a cell is separated into two? How can different amino acids from protein crack lie? The answers are all in this little thing, DNA. It's actually a giant guy in the molecule family with a giant formal name, deoxyribonucleic acid. When we tear this name apart, we can get to know a little bit about DNA. Deoxyribo means it lacks oxygen, but there's a misconception that DNA contains oxygen. It just has one oxygen less than RNA in each monomers. Nucleic acid. When you see this word, if you associate them with nucleus, then you're on the right way. Because DNA, deoxyribonucleic acid, is a molecule that carries the genetic instruction used in the growth, development, functioning, and reproduction of all living organisms. Every part of you depends on DNA. Your outfit, hair, face, your blood type, A, B, O, and AB. Your characteristic, no matter you're outgoing or quiet. Your sexuality, your ability in sports. Basically, your body is full of DNA. They are in every cell of your body. How essential DNA is for you and all forms of life. They are in the nucleus and too small for you to see. However, you are lucky today. A DNA lady just dressed up. This is DNA. Looks like a twisted ladder. Oh, watch out! The shape of DNA molecule is a double helix. When your side is straight to the molecule, the little steps of the ladder seems like many lines, and you may see this picture of DNA on textbook. But when you observe it from the top, it becomes this or this. You're finding donuts. There's actually sugar on them. The sugar is called deoxyribose. That's where the name of the DNA molecule comes from. Our cute donuts are 4 different nitrogen-containing nucleobases: adenine, thymine, cytosine, and guanine. Now, there's only one part of the molecule missing, a phosphate group, which adds flavor to the donut. The deoxyribose phosphate group and one of nitrogen-containing nucleobases link together by covalent bonds. 2 nitrogen-containing nucleobases link to each other by hydrogen bonds instead of covalent bonds. Do you remember how you felt of ladder? Because DNA molecules are different from other micromolecules. There are 2 biopolymers twisted together. They are not sharing electrons due to that, so links between them are weak. And these 2 strands run in opposite directions. Each has a 5' end and 3' end in opposite direction as also. In every strand, the place where there is phosphate group is 5', and the place with hydroxide group is 3'. Not only the strands have the 5' end and 3' end in every unit, comonomer has 5 and 3' ends. All the strands naming system of this part is based on which carbon atom is bonded with our donuts and phosphate group. Number 5 carbon likes phosphate group, and number 3 carbon is a foodie who likes donut. In the DNA molecule, hydrogen bonds between nitrogen-containing nucleobases also have a strict linking relationship. Adenine can only attach to thymine, cytosine can only connect to guanine to form base pairs. But why? There are 2 love phrases. First, let's go back to the donut picture. You can see that A and G both are 2 donuts, and T and C are structured with 1 donut for each. So if A and G are coupled, and C stays with T, then the part of the chain will neither be too wide or too narrow. The DNA lady won't be as beautiful as now. Second reason is more serious. The way the donuts form only to ability to form hydrogen bonds. A and T both have ability to form 2 hydrogen bonds. Meanwhile, C and G can all form 3 hydrogen bonds. Then why don't they get together? Another thing about DNA molecule is that they're really long. One DNA molecule is about 3 billion bases. Let's assume that you're climbing a DNA ladder. It will take you almost 350 years to climb to the top, if you don't stop at all. If you want to achieve this goal, make sure you have already convinced your grandchild to help you climb the rest of the information ladder. Now, after a long talk with your future offspring, you know how big a DNA molecule is, And you also know that DNA molecules are in every cell of your body. So how mother cells making sure the daughter cells have the same DNA? Does it chop itself into two, or copy it like you do to a book? DNA is actually smarter than all of us think. The process is named as DNA replication, and it's semi-conservative replication because when the process finished, you can see the two new molecules both have a chain from its mother molecule. Beginning with helicase separates twined DNA strands into two single chains. It's the same movement as you unzip your clothes. The separated part is a replication fork. Now, there are two templates for making two new DNAs. It doesn't seem very complicated, isn't it? The next question is when to replicate. Let me introduce primase to you. Like a start button, a primase starts the process by letting bases finding their right positions to form a primer. Primer is an RNA. The C and G are still good couple, T with A, but A linked with U instead of T. Primer is starting point of building new DNA. Then, no matter you're playing with toy bricks or you're engineered to build buildings, you always have to make your first step. How about red block? After your primer is built, you don't want to do the rest of the work by yourself, so you call a helper over, DNA polymerase. You attach the DNA strands. It works very hard. T with A, C with G, C with G, A with T. It doesn't stop. One of your DNA strands is full of well-arranged bases on the other side, which means you replicate DNA molecules successfully. Unfortunately, your helper can only help you to build your information grid wall in one direction. You must remember that DNA strands run towards opposite ways. Also, the replication can only start from 5' end and finish at 3' end. You and your helper have to work harder. He builds another primer at here. The replication continues, but if it reaches the earlier primer, the process stops. The DNA polymerase has to build another primer to let the replication go on. The pieces of DNA between primers are Okazaki fragments. After about an hour, this strand is full. So I bet you're wondering why these two new molecules are different from each other. The reason is that they are not finished yet. The exonucleus will come to break the primers, and DNA polymerase places bases to DNA chain. For the last process, the DNA ligase come to make two new DNA molecules continuous. It seems like you revised your essay, taking the wrong part out and put the right part in. Now you have two new DNA molecules. DNA replication is hard work. However, we finished doing today. And all of the process is going on just right inside you now.